Okay, guys, so we finished with replication, right? Yeah. So please remember replication. Now, there's two types of replication: linear chromosome and circular chromosomes. Circular chromosome replication is not a problem. It's bidirectional. In linear chromosome, it is bidirectional only in the linear way, not in circular way. Now, in circular chromosome, you don't lose any nucleotides, correct? In circular DNA, when you copy, you don't lose anything. There's no chance because it's not open. In a linear chromosome, when you copy, sometimes the ends can be erased, can break, things can happen. And this is usually associated with aging, believe it or not. The end of this chromosome are known as telomeres. Telomeres. When a chromosome divides, the ends, you have to add a lot of nucleotide called telomerase. The enzyme will do that. With aging, we lose the ability to add telomeres to the end of linear chromosome. So only linear chromosome can actually age. It's crazy. Mitochondria is not supposed to age. Okay? Bacteria is supposed to not age. But with them, problem is they burst because of pressure and other things. But as far as DNA is concerned, linear chromosomes are more at risk because you can lose from each end. And when you lose, there is less DNA, and the likelihood of producing the same product decreases. So one more thing, linear chromosome. At each cell division, linear chromosome should actually be elongated to maintain the same length. Okay? Okay. I don't know if there's anything else that I remember what you said. Okay, DNA polymerases. Very good. There is something. DNA polymerase. Now, these are extremely important enzymes. Let's compare them to RNA polymerase. So, they're known as DNA pol, RNA pol. Okay, I want to differentiate between them. I want to compare them, and then I'll come back to the types of DNA polymerase. Now, number one, DNA polymerase, they only make DNA, right? They will make DNA. That's all they do. They cannot make RNA. Yeah. RNA polymerase only make RNA. They cannot make DNA. Okay? Yeah.
RNA doesn't make DNA.
What is it? Oh. RNA. There you go. My tongue and my brain don't work together. Everywhere everything wants independence. It's crazy. Yeah, everything wants freedom. And guess what? Energy makes you a slave. The day you let go of energy, you're free. Okay, now, so that's one thing. Now, DNA polymerases, remember, they only recognize what? Deoxy. They recognize deoxynucleotide, and RNA will recognize ribo or oxynucleotide. Okay. DNA polymerase do not recognize, don't recognize U to be normal. If it comes across U, it removes it. RNA does not recognize T. If it comes across T, it removes it. It's a mutation. It shouldn't be there. It does not belong there. Now, there's something unique about RNA polymerase. RNA polymerase is a much larger enzyme. RNA polymerase is much larger enzyme. DNA polymerase is slightly smaller. And at least in terms of function, not in structure. Structure, I don't know what they look like exactly. So I'll be honest with you, I don't know. If you want to look for it, go see if there's some facts regarding that. DNA polymerase only has polymerase activity. Polymerase activity. It only has polymerase activity, which means it only combines DNA nucleotide. It only combines DNA nucleotide, and the 5 to 3' direction. That's all. That's all it does. It makes new DNA. It can only combine DNA nucleotide. That's DNA polymerase. RNA polymerase has polymerase activity. Of course, it has to. I mean, that's why it's called polymerase, right? In addition to that, it has helicase activity. It also has single-stranded binding protein activity. So, RNA polymerase has intrinsic helicase abilities, intrinsic single-stranded binding protein abilities. So, when we talk about transcription, you no longer need helicase, DNA helicase. You don't need single-stranded binding protein. And you don't need primase, because RNA polymerase can do all of that by itself. Okay? All right. Let's talk about types of DNA polymerases. Now, in order to understand that, you should know, again, all of us have gone to school. All of us are exposed to some information regarding different lives, different type of cells. Especially I'm talking about eukaryotic cells. Or eukaryotes, eukaryotic cells, but let me just say eukaryote, that'll be easier. And pro. What does karyo mean, karyo? Nucleus. Good job, nucleus. What are some changes that happens in necrosis? I just remember I didn't talk about it. So I'm asking you guys. No, in necrosis, what are some changes that occurs within? What are the terms for that? Pycnosis. Very good. Karyolysis, the nuclear membrane dissolves, disappears. Very good. Karyorrhexis, the nuclear disappears and gets fragmented. Excellent. So karyo means nucleus. Okay, so please, you had it in PowerPoint, I hope, right? Amazing. Good for him. Good for him. Okay. Yeah, my mind is wild. I just, whatever it goes, has something to forget. So karyo means nucleus. EU, why is Europe called Europe? True. European Union, at that time, they said, we're the true people. Everybody else is not true. So U means true nucleus. These cells have true nucleus. Pro do have nucleus, but it's not true nucleus. It's before the cells. So for example, eukaryotic cells should have a nucleus that is separated by a membrane. A membrane. Prokaryotes don't have a true nucleus, meaning it's not separated by membrane. You can call the center of the cell a nucleus, no problem. But it doesn't have walls around it. It's not separate from the rest of the cytoplasm. Prokaryote is just an ocean of cell. There's fluid and then there's circular DNA floating around somewhere. And the prokaryote you have to know is what? Bacteria. You don't have to know any other prokaryotes. I don't know. There's only two types I know is bacteria and there's those high pressure bacteria. What do you call them? Oh, there's a term for it. Old. What is old? Archaebacteria. There you go. Very good. So anyhow, now we're going to talk about pro and eukaryote. What was I talking about? Why did I even say that? Polymerase. Thank you, thank you, thank you. In eukaryotic cells, which is human, worms, okay? Everything except bacteria and viruses. Every other cell is called eukaryote. Bacteria is prokaryote. Viruses are what kind of cells? They're not cells. They're not cells. They're not living. So eukaryotes will have polymerase. Okay, polymerase, DNA polymerase. Let's just put here DNA because we're talking about DNA polymerase. It's called alpha. Beta. What's after beta? Delta. What's after delta? Epsilon. What's after gamma? I think gamma comes between B and, right? Beta and data. Okay. Let me do that because my Greek friends might not like that. Alpha, beta, gamma, delta, and epsilon. This represents A, B, C, D, E. That's what it technically is. Okay. And prokaryote, we don't have these types. We have DNA polymerase 1, 2, 3. Good? Is it in your book? I'm glad it is. Okay. Alpha is primarily for making the lagging strand. Alpha, this polymerase makes the lagging strand. Lagging strand. Lagging has LA, A for lagging strand, A for lagging strand. Now, D for leading strand. D for leading strand. Or delta. Are we good? Alpha is done, delta is done. B for bad strand. Is there such thing as bad strand? There's possibilities. And when you have a bad strand, what do you do with it? Repair, get rid of it. So they do repair. Beta will do repair, repair. Excellent, excellent, excellent. Epsilon makes bad strand excellent. so it also does repair. Or excision. Excision. I don't know. For me, mnemonics help me a lot. I don't know if it does for you. A or alpha for lagging. D for leading. All the epsilon can do some leading as well, but it does repair mostly. E and B does repair. Gamma is unique in a sense. I should have done this put by data. Gamma is only in the mitochondria. Gamma is only in mitochondria to make mitochondrial DNA. Mitochondria has its own DNA, right? Okay. In prokaryote, which is bacteria, we don't care for it now. I just want to remind you there's such thing. I don't want to confuse it with that because next semester, what are we going to do? Bacteriology, virology, and parasitology, and mycology. We're going to do all the immune system and infectious diseases. Good. So please, this is important that human beings have alpha, beta, gamma, delta, and epsilon type of polymerase. Each of them have specific function. Okay, proofreading is done by, for example, by alpha and beta, but then when you talk about, alpha and delta, when you talk about beta and epsilon, they do repair. They'll repair those gaps. They'll fill it in. Okay, all right, good. Okay, what else? Let's go by the book, so because I think I'm forgetting stuff in between. Yeah. What is it? Yeah, 12, 15, 12. Let's do transcription. Let's do transcription, and that should at least give us some head start for Friday. Okay, transcription. So we did replication with transcription. Transcription is when you take DNA. When you take DNA and you copy it into mRNA. That's transcription. Okay. Or generally RNA. Transcription is copying RNA from DNA. Could be ribosomal RNA, tRNA, or mRNA, but mostly mRNA. That's transcription. Okay. What is reverse transcription? Going from RNA to DNA. Human beings have no abilities to do such thing. Very good. Human beings cannot do reverse transcription because they don't have the enzyme known as reverse transcriptase. Human beings cannot do mRNA from DNA. DNA from mRNA, sorry. They cannot do the reverse of transcription. They can do it. Some viruses can do that. For example, HIV virus. HIV virus. HIV virus, yeah? Okay. All right. Okay, let's start with that now. So you have a double strand of DNA. Here is the enzyme known as RNA polymerase. It's a large enzyme. This enzyme has the ability to do almost everything by itself. Because it's large, it has a lot of area to cover, so it does the helicase function by itself. It will do the helicase activity. So number one, it has the helicase activity. Okay. It It will also stabilize the DNA. Now behind it, the DNA will close spontaneously. As you make the new RNA, let's say the new RNA is in red, as you make the new RNA, it will just keep falling from it, and then the DNA behind will keep closing, and if it's going in this direction, you copy and copy, copy. At the end, DNA will come back to double strand, and then you just made a new copy. A short sequence though, you don't copy the entire chromosome. See, number one, we have to understand transcription is only for what? For protein synthesis. I should have talked about that. What is the purpose of making mRNA? To make proteins. To make proteins. Okay, to make proteins. Let me erase this. I should have done that first. So taking DNA and making a photocopy of it in the form of mRNA is extremely important. And the only reason you will want to make a photocopy, so once you're done with it, you don't care for it. You can shred it, you can throw it, you can burn it, whatever you want. But as long as you get something out of it. Now, proteins, DNA is located where? Within the nucleus. Remember we said that in the nucleus you have these large pores. DNA should not be able to leave. RNA can leave. Now, protein synthesis occurs where? Cytoplasm. What are the machinery or factories who make proteins? Ribosomes. As far as we know, ribosomes are not located in the nucleus. They are made, sure, assembled, yes, but they're located where? Functional ribosomes. In the cytoplasm. Some are free, some are attached. So, the code for protein synthesis is located on DNA. And the factory that makes, so let's say this is the large and this is the small subunit, the factory that manufactures, makes protein is in the cytoplasm. And the code is where? In the nucleus. So you have to copy that code, make a photocopy of it in the form of mRNA, and then it goes to this channel, it goes out, and then it comes to the cytoplasm where it's translated into protein. So therefore, when we want to make a photocopy of mRNA, we have to do it right, of DNA. So the whole purpose of transcription is what? To make a photocopy of DNA so you can use it to manufacture proteins. Protein synthesis occurs where? In the cytoplasm. And DNA is located in the nucleus. Now, why don't you bring DNA directly into the cytoplasm? Very good. Yeah. In the cytoplasm, you have enzymes known as what? DNAs. You also have what? RNAs. And the specific types of them are known as endonuclease. And the nucleus have these activities. And you have another type known as exonuclease. Write this down if it's not in your book. I don't think it is. Exonuclease. Endonuclease and exonuclease. Let's see what's the difference. This is a linear molecule of nucleic acid, DNA or RNA, doesn't matter. If you can only cut, you know what, let's just do this. Let's do one red and one green to make it easier. Now, when you cut only from the end, that's what exonuclease can do. It only cuts one nucleotide at a time from the end. Exonuclease, exo, outer. End. Exonuclease can only cut one nucleotide at a time from each end. Whereas endonuclease can cut within. So the end result is exonuclease gives you single nucleotides, right? Because it can cut only one nucleotide at a time from each end, let's say. So exonuclease will give you multiple single nucleotides when it cuts the DNA into fragments or into nucleotides. Endonuclease do not cut from end. Where do they cut from? The middle somewhere. Let's say you cut at the position green. Let's cut right here. Let me use this. We cut right here. And then you cut here. Are you getting single nucleotide now? You're getting fragments of nucleotides. You're getting fragments of nucleotides. So that's the difference between endonuclease and exonuclease. Now these guys are found primarily in the cytoplasm. In the cytoplasm. And if you send your original copy, even though it's stable enough, you can protect for a while. But if it comes to the cytoplasm, so you can use it to make one protein. Are you going to take the entire chromosome out? You know, chromosome has multiple genes on it. And you just want to take one gene and translate it. That's crazy to take the whole chromosome, which is huge. You know, the length of a chromosome is 10 times longer than the diameter of the nucleus. But it's condensed, so it fits in there. It's crazy. So first of all, it's not practical to take the whole chromosome for a small chunk of it to be translated. Why would you want to take the whole? Let's say I tell you, you know what, this semester we're going to only cover, from Robbins pathology, only the genetic diseases, which is 50 pages. But the book is 1,500 pages. You're going to say, you know what, I am a smart student. I'm going to photocopy the entire 1,500 pages, even though I want you to do one page. It's expensive, it's useless, it's not a good idea. So that's why you don't want the entire chromosome to come out. Even if it comes out, it's not safe. It's not safe environment because your book is probably made of gold. Somebody might take it away from you. Okay, so it's not safe. Therefore, you just want to make a copy of the region that you need only. And that makes sense. Why do you have to copy the entire book if you just need one chapter, one page? That's what transcription comes. Okay, so therefore, when we do transcription, we're only transcribing that single gene, unless there's two genes together. Sometimes you can do that. And it's a small chunk, no big deal. So mRNA is small compared to a huge chromosome. It can fit through this channel. So first of all, it can come to the cytoplasm. Even if it comes once you get your product, you don't want this to be there forever. So you can cut into pieces, shred it, no big deal. You got your job done. Next time you can make another copy, another photocopy, another photocopy, another photo. No big deal. And you keep digesting it. But if you have DNA to cytoplasm, it's a problem. Okay, so that's transcription. Let's move on now with the process of transcription. Steps in transcription. Well, first thing is DNA has to be uncoiled. Still top isomers would have to do that. Okay. Now, if it's a large gene, you will need uncoiling. If it's small gene, you don't need uncoiling. Because large genes has the probability of twisting. A long rope can twist small area. Let's say this is chromosome. Remember, chromosome is usually condensed. This is inactive. So this is inactive. So first you have to open that region. You have to open that region. Okay, once you open it, then you have to unzip it. Only the opening requires that enzyme. As far as the unzipping and transcribing and everything else, you need one enzyme, and that is RNA polymerase. Okay. So let's say we have DNA that's already in the linear fashion, and I'll tell you the rest later. It's already in the linear fashion. Now RNA polymerase will come, and it'll make a cut in the center. It will go in here, and it will start cutting. Okay, so the nucleotide, unzipping the DNA. At the same time, it can stabilize it. At the same time, it can copy it. It can open it, copy it, and stable. All three can be done by one enzyme, and that's RNA polymerase. Okay? So it's a very straightforward process. Transcription is much easier. All you need is RNA polymerase, RNA polymerase, RNA polymerase. And there's no need for hybrid formation. There's no primase needed. So the things that are not needed, let me write them down. Things we don't need. We don't need DNA helicase, because RNA polymerase has that ability. We don't need, so here let's put don't need. But there's gene expression is a little bit complicated. I'll tell you that soon. No need for these. So DNA helicase, we don't need. And then do we need single-stranded binding protein? No, we don't need them, because DNA polymerase has that activity. It can do it on its own. It can do this, and it can do this. Okay? All right. Primase. Do we need that? No, we don't need that. Should I put crosses then? Yeah, there you go. We don't need these. Primer. Addition of primer. Do we need that? No. So transcription does not require these things, because RNA polymerase can bind single-stranded DNA, no problem, has no problem. Okay, we don't have those problems. Okay. All right. So that's for this. Just give me 10 minutes. Hopefully I can finish it. Now, in order to do that, we have to understand something. there has to be something for a gene. The RNA polymerase should have some sort of signal for it to recognize a gene. Because DNA sequence is a huge linear sequence of nucleotides. Where do you start? What if you only want to translate or transcribe, for example, insulin gene, glucagon gene, or lactase gene, or fructokinase gene, or whatever gene? You're not going to copy or translate genes that you don't need. Why make things that you don't need? You only will make things that you need. So let's say we have a linear sequence of DNA, huge. And here is a gene. Do you guys know any gene? Or any product, any protein? Huh? Very good. So let's say this is one gene, and then there is another one. Let's make it Bluetooth. Okay. So gene 1, I'll call it G1. Gene 2, gene 3. Let's say this one codes for insulin, and let's say this codes for its opposite, glucagon. These are peptide hormones, glucagon. And let's say this codes for an enzyme. In this case, let's say trypsin. Okay. Now, you only ate carbs. You don't have any protein. So why would you want to make trypsin? You don't have to break protein at this point. You don't need trypsin. You only ate sugar, let's say. So glucose. You need insulin for that to transport it. So this gene, you only want to activate which gene? G1. Each gene should have upstream. Upstream means before it. Okay, so this side is, the left side is upstream. Right side is downstream, meaning the direction of information flow is this way, from left to right. Okay? Now, before each gene, upstream of each gene, you should have promoter. Promoter. There's one promoter, there's another promoter. Each gene will have its own promoter. Okay, promoter. Just write it down for now. Each gene should have a promoter. As a matter of fact, between promoter and the gene, you should have operator. Green is operator, red is promoter. And then let's make a yellow. Complete my rainbow. And let's call that inducer or repressor. Okay, so green was what? O, operator. Red was what? P, promoter. And then this yellow part is either inducer or repressor, depending what. Okay? Let's just call it regulator. There you go. That's much better. A regulator can repress something or induce it. Some of you probably seen this in your universities and colleges, and it was almost like lac operon, tryptophan operon, if you talk about, like, you know. But guess what? Each promoter, so promoter, let's talk about promoter first. In the center, I'll make it, you know what, I'll make it larger so I have more room. Let's make this large sequence, and let's make this slightly smaller, and then blue, there you go. Let's say we want to make glucagon, forget it, you're just hungry. Right now you're hypoglycemic and you have to make glucagon. Yeah. Operator. You're welcome. Operator, promoter, regulator. Are we good? There you go. Okay. Let's ignore the other genes, because at this point, look, insulin gene will be activated by too much glucose. Insulin gene has to be activated when whatever you need. The purpose of insulin is to take up glucose into cells. So when you have too much, insulin gene should be activated. Glucagon is needed when you have no glucose. So it should be activated by lack of glucose. The two cannot be activated at the same time. Makes no sense. Insulin is produced when you have too much glucose, so you want to store it. Glucagon is released when you have no glucose, so you can make new ones. When your blood glucose level is low, you need glucagon. When it's high, you need insulin. They're two opposite. They cannot be made at the same time. Makes no sense. You don't need to do that. When you have low blood glucose level, you need glucagon. When you have too much blood glucose level, you need insulin. Insulin removes glucose from blood and stores it in cells. Glucagon breaks glycogen to increase blood glucose level. So huge difference. Insulin wants to store glucose. Glucagon wants to release stored glucose. Break it. Make sense? They do the opposite. Okay. When would you need glucose? When you're hungry, when you're studying for exam, your brain needs more sugar. So the glucagon. Let's say at this point you're stressed, you want to understand this, and it's glucagon. Okay. Now in this case, this gene usually you know what? Here you go. The operator region. The operator region is technically almost like a brake. It will bind something that will prevent transcription. Let's say this is called an inhibitor. I'll put an I. Or let's just, yeah, I don't know what to use. The terms, all terms can be used, that's fine. There you go. This dark thing, let's say, is an inhibitor. You could call it inhibitor, or you could call it repressor. But the letter might make you confused because we have R already.
How about the regulator?
Regulator can be both. The gene regulator, it's a gene. Okay, let me tell you first this one. Okay, operator is nothing more than a sequence of DNA. Operator is a sequence of DNA. Let's define I'll define it first, and I'll tell you how it works. Operator is a sequence of DNA that binds a repressor or inducer, one of them. Usually repressor. Operator is a sequence of DNA, nothing more. And it's always upstream of the gene, before the gene. Operator is a sequence of DNA, a short sequence of DNA, few nucleotide in length. Operator is a sequence of DNA that usually attaches or binds a repressor. Repressor. It binds a repressor. Okay, that's operator. What is it? It's a sequence of DNA. Operator. Let me just tell you. Operator is a sequence of DNA upstream of the gene, whatever gene that might be. Operator is a sequence of DNA upstream of a gene. Its purpose is to bind repressor. Its purpose is to bind repressor, primarily. In rare cases, it can bind inducer as well, but mostly repressor. Repressor. Good? Promoter is a sequence of DNA, again. It's another sequence of DNA. See a region on DNA. This region you call it something. You call this region something else. You call this region something else. That's all. So promoter is a sequence of DNA that binds polymerase. P for P. Polymerase. Which polymerase? Only RNA, not DNA. Only RNA polymerase. Promoter is a sequence of DNA that only binds RNA polymerase. Make sense? RNA polymerase, good. Regulator is a sequence of DNA, but it's actually a gene. See, promoter and operator, they're not a gene. They're non-coding regions. They're just a region which binds something. Regulator is actually a gene, and the gene will code for either inducer or promoter. And in this case, inducer and promoter will be a protein. So regulator is a sequence of DNA or a gene that codes for inducer or promoter. Once you understand this terminology, life will become a little bit easier. Just a little bit easier. Okay. Okay. Now, let's say we have, in this case, polymerase. Yeah.
It's a sequence of DNA. Shouldn't that be an RNA because we're talking about an RNA?
No, we haven't gone there yet. We're trying to make mRNA. So we haven't started transcription yet. Okay, so the idea is how do you make a transcript? I mean, remember, on a single sequence of, excellent question though, on a single chromosome, how many genes could you have possibly? A lot. Many. How many of them are you going to transcribe? Whatever you need. You're not going to transcribe the whole thing, so you better have some sort of mechanism to only activate a single gene when you need to. So each gene should have some regulatory sequences before them. Because if you have, look, now let's say we have RNA polymerase. RNA polymerase will transcribe any gene. There's no specific RNA polymerase for each gene. That's not true. As far as I know today, maybe tomorrow something. Currently, RNA polymerase can activate any gene they want. But first they have to be able to attach to it and recognize it. If the gene is not exposed, it's covered, it's hidden, they won't be able to attach or copy it at all. That means there's a mechanism. So let's say this is RNA polymerase. I'll just make a, let's say this is RNA polymerase. See, RNA polymerase swims around randomly all over the place. And you see this gene is covered. Okay, this gene is covered, this gene is covered, they can't attach. This gene is covered, it cannot attach. That's first thing. So when you have a need of something, there's something known as transcription factor. Remember we talked about this? Transcription factor. So transcription factor will attach to what? To RNA polymerase and to the promoter. Therefore, exposing the exposing the promoter. Now, RNA polymerase can come and attach. Okay, they can attach. Not promoter, not exposed unless you need them to be. Unexposed promoter cannot be transcribed. Transcription cannot start unless RNA polymerase is attached to promoter. Now, promoter is the sequence of DNA, not RNA, DNA, where initially transcription begins, where RNA polymerase will initially attach. They cannot attach anywhere they want. That's the only place they are able to attach. And the sequence of the promoter is conserved. It's known. It's known as the Tata box, those of you who know. Tata box. This is the sequence of promoter. It's a DNA. So RNA polymerase always recognizes Tata box, but it has to be open and exposed. And who does this exposure? Hormones, transcription factor, and we talked about those signal transduction. You have to go through all of those things. For example, if this is glucagon, you know that glucagon has to attach to receptor and go through cyclic AMP, protein kinase A, transcription factor, then you get to here. Okay. So now, first, RNA polymerase can do transcription, no problem. But they have to attach first to a sequence of DNA. And the region where they will attach is known as the promoter sequence. Because if they attach in the center of the gene, how much would they copy? Because copying is only one direction, half, and that's not right. You get only half a protein, and that's mutation. So transcription must start before the gene starts. So anyways, promoter is a region of DNA. It is a region of DNA, a sequence of DNA upstream of the gene where RNA polymerase will attach. Where RNA polymerase will attach. But think of it as like an airplane. The airplane cannot land if there's cars parked on the highway, or the air, what is it called, the air, yeah, or runway. They cannot park there, so you have to remove and clean, get rid of the snow, so then they can park. Same thing with RNA polymerase, and that requires a lot of processing. But the runway is only a short sequence of cement or pavement. You don't use, for example, Lagrange for landing your plane, right? So that region, that sequence of DNA where polymerase will attach and land is known as Tata box or promoter. Okay. All right. Now, so that's good. Now, what if there is something in front? So repressor, I'll use red. Repressor is a protein that attaches to promoter, and it blocks the progression of polymerase. That's what the repressor inhibitors do. Repressor protein. We're not talking about a gene anymore. Repressor protein. The one in red. This repressor protein. It literally sits in front of the promoter. It can only attach to a sequence that's in front of the promoter, and that sequence is known as what? Operator. Operator. So repressor can only attach to the sequence of DNA called operator. When it does attach, it blocks the progression of polymerase. Well, polymerase can attach, but if there's repressor, they can still not do anything. Okay. Now, regulator can remove, actually, the repressor, the product of regulator, depending if it's repressor itself or its inducer. Okay. In this case, usually glucagon should be blocked. The synthesis of glucagon should be blocked. When you get hungry, when you need glucose, then lack of glucose can be inducer. Or, in this case, let's say cyclic AMP. Cyclic AMP. So cyclic AMP, for example, comes and it will attach. Let me use purple or dark. Cyclic AMP comes and attaches to what? To the repressor. And then, empty repressor. Empty repressor can attach to the operator. Occupied repressor cannot attach to operator, for example. So as soon as you have cyclic AMP and it can attach to repressor, and then the repressor will move what? Out of the way. And now your RNA polymerase can initiate transcription. And it will copy the genes downstream of its initiation. Okay? A lot of terminology, right? A lot of names. But you just have to calm down. I don't know if you guys ever seen those VHS videos, whatever they call it. The tapes that used to run. This is the same thing. Same thing. I guess it helped me when I was a child. I used to break and try to make things out of it. Now there's CDs. I cannot give you those examples. But it's the same thing. It has to scan step by step. Okay, so first it has to attach, and then you have to remove the repressor, and then it can proceed. It's almost like opening it. You know, first you turn on your car. You have to go sit in the car. You have to sit in the car, and that is, you know, polymerase have to sit in the car, and that is promoter. Once you sit in the car, you turn it on, and then you have to open your garage door, and that is the repressor. As long as the repressor is closed, you can't move, and then you can start copying. So this is required. All genes. This is gene expression. Make sure you know promoter sequence. You have to know the title box. Make sure you know what's operator. Make sure you know what's regulator. Make sure you know what's polymerase, and you make sure you know what is inducer and repressor. Okay, if you have too much inducer, gene will be active. It will be transcribed. If you have too much inhibitor or repressor, you will have inactive gene. So it's always a balance between them. For example, let's say you drink milk, a lot of it, which means you need a lot of lactase enzyme. So that means you have a lot of inducer, and the inducer is what? Lactose. But once you break lactose into what? What does lactase do? Yeah, so those of you who don't know, that's fine. Lactose is a combination of glucose and galactose. Lactase enzyme has to break it. Now, when you drink milk, you need the enzyme to break it so you can absorb it. Now, before breaking, lactose is an inducer of this gene for lactase. Because it tells you, I'm here, I need you to break me. So it has to be an inducer of lactose gene. But when you break it, what do you get? What's the product? Glucose. When you have too much glucose, it's a single signal for what? That's one thing, yes. It's the signal for the same gene. You have done enough production of lactase enzyme, stop. Because I'm the product, I'm here, I'm the result of that breakdown. So the product of a reaction is always inhibitor. The reactant is always inducer. Okay. If you don't understand, let's go, and because I have to start that one again. Let's take a break, and I'll do something on Friday. I'll do slower. Just read a little bit. Come prepared on Friday. It'll make much more sense when you read something. Okay.